In section 8.2, I'm going to start off just reviewing some things about DNA and RNA. And like I said, if you're really rusty on this, you might want to go back and do some review using videos on the internet. But this should be a pretty quick, useful video for those of you that just needs a little bit of reminding. So remember, we said all cells have DNA and RNA. We said viruses have DNA or RNA, but all prokaryotic cells and eukaryotic cells have DNA and RNA. And probably most of you remember that DNA is a double helix. We've got this kind of helical structure. And along these, if we look at this as a ladder that's been twisted, we can look at the sides of the ladder. And here it says a helix of sugar phosphates. So DNA is called deoxyribonucleic acid. That's what DNA stands for. And so along these sides of a ladder are sugar and phosphates. The deoxyribose, right here, is the sugar. So we'll have a sugar and a phosphate, and a sugar and a phosphate, and a sugar and a phosphate going all the way up and down each side of this ladder. So DNA is named after this 5-carbon sugar that's found in this molecule. And then remember, you have the rungs of the ladder, the steps of the ladder, that are the nitrogenous bases, or here they call them nucleobases, where adenine and thymine come together, and guanine and cytosine come together. So you should be a little bit familiar with that structure. But the main reason why I'm even showing you that is I just wanted to remind you about deoxyribose being in DNA, because that's going to be important. RNA is very similar to DNA, but RNA is a single-stranded molecule. You can still see it's still kind of helical, and it's got bases, and it's got the A, G, and C, but instead of T, it has U. So you might remember that from some class that you had at some time. And so there we have RNA, and the side, this helical part, is made up of a sugar phosphate. But the sugar happens to be it says RNA is called ribonucleic acid, so the sugar is ribose. So I want you to notice here that deoxyribose in DNA and ribose are really similar, except guess what? There's a deoxy. There's a minus an oxygen in deoxyribose. And this is going to come up in a few minutes when we talk about, in another video actually, when we talk about mutations. So keep that in mind. The other kind of overview of information I wanted to remind you of is just the concept of transcription and translation. So it says provide an overview of the flow of genetic information and the relationship among DNA, RNA, and proteins. Now this term right here, flow of genetic information, that's what I'm going to describe here, how DNA is used to make RNA, RNA is used to make proteins. Some places you look, they will call this the central dogma, meaning it's a central idea in biology. Dogma is like an accepted fact, accepted idea. So central dogma. So you might hear that in biology, when cells use DNA to make RNA and use RNA to make proteins, they might call it the flow of genetic information. They might call it the central dogma. And your book uses a term I don't usually see people use, but they have the term the central theme in biology, that DNA is used to make RNA, is used to make proteins. All of those things are asking you the same thing. I'm sorry that's so confusing, but that's the way it is. Okay, so we want to talk about this flow of genetic information, or the central theme in biology. And I'm going to give you an analogy. If you remember all this already and you're good with it, this might be too simple, but here you go. Just to remember this, I think about, we have our kitchens because we like food and we want to eat food, and food is what drives us every day and gives us energy. Cells they have DNA, they have an RNA, but the reason why they have those is to make proteins. Proteins are what runs our cells. They're the enzymes, they're the structural molecules. So I'm going to make an analogy between making food and making proteins. So if we look at our recipe book, I have a cookbook. I have all sorts of cookbooks. But here's my recipe book, my cookbook, and it has all the instructions to make all the foods that I like or that I need. And we can look at DNA in that way. DNA has all the instructions to make all the proteins a cell needs. So what the heck is the RNA doing? Well, if DNA is all the instructions, that's the genome, remember that? Every once in a while, I take out my cookbook and I make a copy. I'm going to make a copy of one page, one recipe, and that's going to be a piece of RNA. So if my cookbook is my DNA, a copy of one page is my RNA. Now remember, we said every cell has every cell in a human body has the DNA on how to make everything in the human body. But you're not making everything all the time. Like we said, our eyes don't make liver proteins. So a lot of those recipes are never tapped into. But when you need to make a certain protein, you're going to copy a page out of your recipe book and you're going to make an RNA. You can imagine a piece of RNA is a lot smaller than a piece of DNA. So we're going to copy a page out of our entire cookbook, and then we're going to use those instructions to make a protein. So if my analogy is my cookbook, my photocopy of a page, and the food I want to make, DNA to RNA to proteins. Okay. So the process by which DNA is used to make RNA is called transcription. And the process in a cell by which RNA is used to make proteins is called translation. Now, you might remember when we talked about cells that we said there was a certain structure in both prokaryotic cells and eukaryotic cells that's the site of protein synthesis. Do you remember what that was? That's where the ribosomes are. Ribosomes are where the proteins are made. So translation occurs at the ribosome. So both bacteria and eukaryotic cells have ribosomes. Okay, so the central dogma, or the flow of genetic information, DNA to RNA to proteins, and DNA to RNA means transcription, and RNA to protein is called translation. So I'm going to build on that. And here we are. It says, explain the concept of retroviruses and give examples of retroviruses. I've added this in the past couple years because I found out that students in nursing hear about this. They talked to them a little bit about retroviruses. So I wanted to make sure at least you heard of this before you got to nursing. But it builds on the whole unique thing about DNA and RNA and proteins. So remember we said some viruses do not have DNA, they have RNA. And so we have a lot of viruses we call RNA viruses. And RNA from most RNA viruses will just attach to a host ribosome, start making proteins, and that's how they make proteins. So if you're an RNA virus, you might just attach to a host cell, get your RNA inside the host cell, and then go to a ribosome and just start making virus proteins. And that happens frequently. But there's some kinds of RNA viruses that are called retroviruses. So retro means going backwards, right? So the RNA of retroviruses is converted backwards. It's converted to DNA. Here we go. Here's our RNA in our retrovirus. It's actually is converted first to DNA by an enzyme called reverse transcript. The enzyme's called reverse transcriptase. Here they're saying you're doing reverse transcription using the enzyme reverse transcriptase. So in retroviruses, the RNA first has to be turned into DNA before the DNA can make RNA and can make viral proteins. And the reason why they talk about this in nursing is because some of the most effective drugs to inactivate retroviruses are chemicals that stop this step. Cells do not need these. Human cells and bacterial cells do not need enzymes to make transcription go backwards, but retroviruses do. And so if you have a drug, and there are drugs that exist that can stop reverse transcription, then you can inactivate those viruses. And you probably know that it's tricky to find drugs to inactivate viruses. And what viruses are we thinking about? Well, mainly we're talking about HIV. The HIV virus, here they're showing you how it attaches to the host cell, gets in the host cell, there's that viral RNA, there's the reverse transcriptase that turns that into DNA, and then the DNA is used to make RNA and make viral proteins. So we go from RNA back to DNA, back to RNA, back to proteins, and now we can make new viruses. So if we can stop this step, if we can stop that viral RNA from being able to turn into viral DNA, you can stop that infection. And that is really the key to some of the most effective drugs in HIV treatment. In fact, since HIV first became, since we first knew about it in like the 1980s, and now it used to be an immediate death sentence. I mean, people lived for a few years, but it was a death sentence. But these antiretroviral drugs are what took HIV from being a death sentence to being something that people can live with in their lifetime, throughout their lifetime. And I just put the example of some of the other retroviruses we know about. There's a cat disease called FIV, feline leukemia, and SIV in chimps and gorillas. All of these are retroviruses, including HIV. And there's another one called human T lymphocyte virus, which isn't as common as HIV. But there's not a lot of retroviral diseases that we worry a lot about, but HIV, obviously in humans, is the big one. I think I'm going to stop right